KRAS, taking on one of cancer research's toughest challenges. KRAS is recognized as one of the most frequently mutated oncogenes in human cancer. However, despite nearly 4 decades of scientific efforts, KRAS has been one of cancer research's toughest challenges. In healthy cells, the KRAS protein acts as a molecular switch to regulate downstream signaling pathways, cycling between a GDP-bound inactive state and a GTP-bound active state. Activation of KRAS occurs following the binding of ligands to receptor tyrosine kinases, such as EGFR, at the cell membrane. When activated, KRAS regulates downstream signaling through interactions with proteins including PI3 kinase, RAF, and RAL. This results in a signaling cascade through other key proteins, including mTOR, ERK, and NF-kappa B, ultimately leading to changes in gene transcription, promoting cellular proliferation, differentiation, and survival. Mutated KRAS may deregulate normal signaling and drive the development of cancers. Approximately 1 in 4 patients with non-small cell lung cancer have KRAS mutations. One specific mutation, KRAS G12C, is the most prevalent KRAS mutation in non-small cell lung cancer and is also found in colorectal cancer and other solid tumors. KRAS G12C is a single-point mutation at codon 12 in which a glycine is substituted with a cysteine. The KRAS G12C mutation impairs cycling of KRAS, favoring the active form of KRAS by slowing the hydrolysis of GTP, leading to oncogenic signaling and subsequent tumorigenesis. Despite being one of the most common oncogenic mutations in human cancers, several hurdles have left attempts to target KRAS unsuccessful, including the lack of surface targets for KRAS binding. However, recent scientific advancements have revealed a possible binding site to target mutant KRAS. In the inactive state of KRAS G12C, the mutant cysteine resides next to a narrow surface pocket, the P2 pocket, providing a potential binding target for covalent inhibitors. Novel covalent inhibitors are under investigation, which are hypothesized to specifically and irreversibly bind to cysteine 12 adjacent to this small pocket of the KRAS G12C protein. Covalent inhibitors may have the ability to lock the mutated KRAS G12C in the inactive form, impairing oncogenic signaling. Irreversible covalent binding of a small molecule to mutant KRAS G12C may be sufficient for prolonged inhibition of oncogenic signaling. By utilizing advances in understanding the protein structure of KRAS, Amgen is committed to taking on one of cancer research's toughest challenges by investigating ways to specifically inhibit KRAS G12C. To learn more, contact Amgen Medical Information.